Alaikum, Bismillah ar-Rahman ar-Rahim. Today we are going to talk about uh, two sensations together. They are called chemical sensations: gustatory, which is taste; olfactory, which is smell. Okay, our objectives is to describe taste and receptors and also olfactory receptors, follow their pathways to the cerebral cortex, and compare and contrast gustatory and olfactory sensation. حكينا إنه both of them are chemical sensation. And also talk some abnormalities of both of them. Okay. Uh, so both sensation, taste and smell, they allow one to separate undesirable or lethal foods from those that are nutritious, recognize the proximity of other animals, and tied primitive emotional and behavior. Usually these, especially smell sensation, is very primitive. It is more developed in animals than human beings. So it's very hard to study smell sensation from the physiologic point of view because it's like subjective things, not objective things. Okay. Uh, if we talk about taste to start with, for taste, let's talk here. Taste substances, taste. They are found in taste buds. Taste buds. This is the taste buds. So taste receptors, they are found in taste buds, and these taste receptors are actually epithelial cells. What do I mean by epithelial cells? Epithelial cells, it means that these cells can regenerate. Okay, so this is very important that these cells can regenerate, and they can regenerate in terms of 2 weeks. They can change altogether. There are too many chemical receptors. Usually we have 5 primary taste sensation. Like when we said about light, we said we have 3 primary colors. We have here 4 primary taste sensations. 13 possible chemical receptors in the taste buds, 2 for sodium, 2 for potassium, and 1 for chloride. These are like salt. Adenosine, inosine, these are for, it's like umami. Sweet, bitter, glutamate, and hydrogen, these are for glutamate for umami, also amino acid, hydrogen for sour, حموضة. We said that we have 5 primary taste sensation. All these sensory receptors are found in taste buds. The taste sensation we have sour, salty, sweet, bitter, and last we have umami sensation. Umami sensation. Okay. So the taste perception is sour can caused by acid concentration, hydrogen, and salty caused by ionized salt like sodium, like potassium. Sweet, many chemicals are mostly organic chemicals. It's not necessarily glucose. It can be any organic substances that feel sweet, like NutraSweet. You know the NutraSweet, they put it on diet soft drinks. This NutraSweet, in fact, is 2 amino acids. One is phenylalanine and the other is aspartate or aspartic acid. So it is dipeptides, aspartic acid, and phenylalanine. That's why you found in the bottles of diet soft drinks, you find that people with phenylketonuria are not advised or are not recommended to drink these things, because phenylketonuria patients, they are unable to metabolize phenylalanine. So this one the NutraSweet contains phenylalanine. Bitter sensation, long chain organic substances containing nitrogen and alkaloid. And bitter substances, usually these alkaloids or organics, usually it is a toxic substance. Umami, not familiar, distinctive, delicious taste that comes from certain amino acids like glutamate and arginine. So these are the 5 primary taste sensations. Where these taste buds are found? These taste buds are found inside the papillae. There are the circumvallate papillae, as they are V-shaped in the posterior surface of the tongue. Foliate papillae located along the lateral surface of the tongue, and fungiform located over the flat surface of the tongue. These papillae, they contain taste buds, extraglossal taste buds on the tonsillar and palate and proximal. There are some papillae that they don't contain taste buds, so they don't contain receptors for taste. Okay, so this is where these primary sensations are located. You see, this is the inverted V shape. This is the dorsal. This is the dorsal third of the tongue here, and the dorsal third of the tongue, or the posterior third of the tongue, it is derived from different area than the anterior 2/3 of the tongue. When you come to the embryology, both the anterior and posterior third of the tongue are derived from different areas. But when the embryo grows, they come together. So the posterior third of the tongue is supplied by different nerve than the anterior third of the anterior 2/3 of the tongue. The posterior third of the tongue has these papillae that's called circumvallate papillae here. So these circumvallate papillae, they have taste buds that are concerned with bitter sensation. And these, that's the posterior third of the tongue here, is supplied by glossopharyngeal nerve. Now the other part of the tongue is the anterior 2/3 of the tongue here. The anterior 2/3 of the tongue is supplied by facial nerve, and it contains the taste receptors that are for sour, for sweet, for salt, and for umami. So for bitter sensation, as you see here, bitter sensation, its receptors are found in the back of the tongue, in the posterior third of the tongue, very close to the pharynx. يعني بدك تفهم انه واي سبحانه وتعالى حطهم هناك ممكن because usually bitter substances are toxic, and if they are toxic you would like to vomit them. يعني بدك تراجعهم. Where is the stimulus for vomiting? الpharynx. فبتلاحظ انه هدول السبستانces, if they come very close to the pharynx, you try to vomit them. So this is where the receptors are located. Okay. Sour in the lateral aspect of the tongue, and the anterior tips of the tongue is sweet and salt. Remember that kids, when they want to lick ice creams, or when we want to lick ice cream, we lick the ice cream by our tip of the tongue. ليش? So as because the sweet receptors are found in the tip of the tongue. Yani we are not going to feel them sweet unless we feel them here in the tip of the tongue. This is the sweet. Okay, so this is why we lick the ice creams or lick sweet substances in by our tip of the tongue. Okay, this is the taste buds here, taste buds, and this is the taste buds. They have the taste receptors, and the taste receptor they have cilia here, hairs. Mm. As I said, the posterior third of the tongue supplied by glossopharyngeal nerve, which is ninth cranial nerve, and the anterior third of the tongue is supplied by part of division of the facial nerve, seventh cranial nerve, seventh cranial nerve. Okay, as you see here, these are the receptors for taste. And these receptors for taste are, as I said, are epithelial cells. They can regenerate. This is where the receptors can regenerate only in the taste and smell. We will come to the smell and show you that also smell receptors, which are actual neurons, they can regenerate, but longer. They take longer time. They take around 45 days. Here, these taste receptors, they take around 15 days. Okay. This is the taste buds. This is the taste buds, has hairs coming from these cells. These are the cells that are the receptors for taste. When we come to taste transduction, the taste substance, the taste ligand, binds to its receptor. Binding to its receptor here causes the signal transduction. By what mechanism? The mechanism is for each substance. Sometimes there is different mechanism, but generally, when the taste receptor, taste ligand, taste substance binds to its receptor, this is the membrane channel receptors, they cause an increase, say one of the mechanisms, they cause an increase in the intracellular calcium by opening voltage gated calcium here. And when there is more calcium, or calcium might also go out from the endoplasmic reticulum, increase intracellular mechanism, increase intracellular calcium by some mechanism, and most probably by activation of calcium calmodulin dependent protein kinase. They cause movement of these vesicles toward the membrane of the receptor cells here, and this they cause secretion of these neurotransmitter that is going to stimulate the primary sensory neuron, taste neuron. Okay. Here the other mechanisms, ligand gated taste stimuli, receptor ion enters, conductance change. Conductance of ions, say it is increase in sodium, depolarization, generation of generator potential or receptor potential. Remember this from last year, causing calcium influx transmitter release. There might be second messenger mechanism, taste stimuli, taste membrane receptor, second messenger. The second messenger might be calcium going from the endoplasmic reticulum or entering from the cells here. And then increase in intracellular calcium cause activation of calcium calmodulin dependent protein kinase, movement of these vesicles that contain the neurotransmitter release to the synaptic cleft, and they go through the afferent fiber of the neuron. Okay, so this is both mechanism can work. Okay, here the different substances and different mechanisms. Like salt substances here, they cause increase in the sodium conductance, and increase in sodium conductance cause depolarization of these cells, and depolarization cause opening of voltage gated calcium, increase intracellular calcium causing. The other sour mechanism here, so the first one is salt, that's opening of sodium channel. The second is sour hydrogen. This binding of the hydrogen to its receptors on the membrane causing closure of the potassium channel. If the potassium channel is closed, is going to cause depolarization of the membrane, and depolarization of the membrane causing entrance of influx of calcium, and increased calcium causes release of the neurotransmitter. The sweet substances, is a G protein receptor, membrane receptor. When the sweet substances bind to its receptor, it causes alpha subunit is dissociating from G protein, going to a membranous membrane enzyme, which is in this case adenylate cyclase. Adenylate cyclase cause the formation of cyclic AMP from ATP, and this cause closure of the potassium channel here. So this is the way. The organic substances, bitter substances, they are like G protein system here. But the G protein, when the alpha unit dissociates, it activates phospholipase C. Activation of phospholipase C causes increase in the inositine triphosphate, IB3. Inositine triphosphate goes to the endoplasmic reticulum and cause release of calcium from the endoplasmic reticulum, and then the increase in calcium causes the same mechanism. So as you see, the common mechanism for all of them is an increase in intracellular calcium. Increase in intracellular calcium here causes the release of neurotransmitter from the vesicles by different mechanisms. Okay, so this is the taste substances. Taste buds. There are more taste buds, of course, in children than adults. Have 3 to 10,000 buds, 3,000 to 10,000 buds. Children have more taste buds. They are epithelial receptors. They can regenerate. By the age of 45, taste buds start to degenerate. Taste becomes less critical. Sweet and salt located in the tip, and sour located in the lateral, bitter located in the posterior third of the tongue. Okay. Transmission of taste sensation, activation of taste buds, their taste receptor, they release a neurotransmitter, activation of taste fibers from the anterior two-thirds through the facial, from posterior two-thirds from the glossopharyngeal. They go here, they go either from the anterior third of the tongue through the facial, called it tympani, from the facial nerve, as you see here, this is the facial nerve, and from the posterior third of the tongue through the glossopharyngeal nerve. From the facial, they go to the geniculate ganglion, and from the glossopharyngeal petrosal ganglion. And there are still other receptors found in the pharynx, and these receptors, they are supplied by vagus nerve. All of them, they go to the nucleus, solitary nucleus in the medulla oblongata and the brainstem. And from there, they go to the VBM, ventroposterior nucleus of the thalamus. From there, they go to the gustatory nucleus, which is found in the insula here, in the insula. Taste pathway is usually bilateral, but predominantly uncrossed. It is predominantly ipsilateral, okay? Predominantly it is ipsilateral, but usually it goes both ways. It is bilateral, okay? So here, posterior aspect of the tongue and the mouth through the vagus nerve transmits to the solitary nucleus, nucleus solitarius in the medulla oblongata, and from there to the thalamus, VBM, ventroposterior median. And you know ventroposterior median, ventroposterior lateral, they are called ventrobasal complex of the thalamus. From the thalamus to the cortex, where in the cortex to the insula. الانسula بتكون قريبة من الarea of the face when we come to the cortex, okay? So, like other sensations, taste do adapt rapidly. In around 1 minute or so, there is around 50% adaptation. Adaptation might be at the level of the receptor, or sometimes at the level of the central adaptation, the level of some central adaptation. Okay. So taste adaptation is one of the sensations that adapt very fast. The other sensation that adapts even faster than taste is smell or olfaction. Smell adapts very fast. Okay, now we come to the smell, the second chemical sensation. The least understood of all sensations, حكينا, because it is more developed in lower animals. It is less. In a human being, it is subjective. You cannot draw a conclusion from these experiments. Then poorly developed in human olfactory membrane located in the superior part of the nostril. Here we talk about this is the superior aspect of the nostril, superior concha. This is where the receptors are located. This is the receptors for smell, the superior concha. لو بتحط نظارة مراية هيك بتطلع على أنفك من فوق بتلاقي منطقة صفراء هناك. هاي المنطقة الصفراء هي receptors for smell, they are located. فشوف هون receptors السميل area. شو في عنا؟ في عنا mucus that is secreted by mucus cells here, basal cells, mucus cells. There are other supporting cells. Then there are other supporting cells here, as you see here. They support the receptors, and these are the mucus cells. They secrete mucus. So you have mucus. The receptors for smell, they are actual neurons. These are neurons, actual. They are bipolar neurons, in fact. And these, although they are neurons, they can regenerate, but their regeneration time takes around 45 to 60 days. They have these cilia. They are embedded in the mucus, and then their axons enter the skull through openings here of the bones called ethmoid bone. So this bone is ethmoid bone, and these axons that enter, they cause perforations or piercing holes, leading to what we call it cribriform plate. Okay. So this is the cribriform plate here. This is the receptors for smell with its cilia embedded in the mucus. Then they pierce or found holes in the ethmoid bone called cribriform, and they enter the brain right away. When they enter the brain, the axons, they synapse with other cells called mitral cells. So the axon, the dendrites of the axon, mitral, the dendrites, of the mitral cells here, and the axons of the olfactory receptor, they synapse together, forming glomeruli here, and each glomerule sometimes is specialized for certain sensations. So the dendrites of the mitral, they synapse with the axons of the olfactory receptors, they form glomeruli. Then after they synapse the mitral cells with the bipolar cells or the receptors, they form what we call it tract, olfactory tract that goes to the cerebral cortex inside. This here just inside the brain, above the cribriform, forming what we call it olfactory bulb in the brain. Okay. Let's go here also. شوف نفس الشيء هون. This is the mucus. These are the receptors, bipolar neurons, enter through the cribriform plate, which is part of the ethmoid bone, and go inside the brain, forming the olfactory bulb neuron. And from the olfactory bulb, they will form what we call it olfactory tract. It is going inside the brain here, inside the cortex. This is the cribriform plate. This is the bone is called ethmoid bone. Okay. طيب هون نرجع شوي نشوف bone here. Bone, ethmoid bone. This is the ethmoid bone. Okay, so this is the pathway, in fact, for smell from the bipolar cells. These are the olfactory gland or mucus that secrete the mucus here. Cribriform enter, forming, these are the glomeruli. These are the glomeruli here, which are the synapse between the bipolar cells and the dendrites of the mitral cells here. The axons of the mitral cell, they form the olfactory tract. نشوف شو نقرأهم كمان مرة نعاود عليهم.